finds the promoter region, the RNA polymerase starts unwinding the hydrogen bonds inside the helical structure of the DNA, and then these two strands are separated from each other. As you can see here, this is the RNA polymerase, this is the winded double helical structure of DNA. Then this RNA polymerase unwinds the helical structure by breaking the hydrogen bonds between these nucleotide bases, and these two strands are separated from each other. Once these strands are separated, then the next step proceeds, which is known as elongation. Elongation, the nucleotides base pairs bind to them at the 3' hydroxyl. In this stage, the two strands are separated. The exposed DNA bases on the antisense strand can now hydrogen bond with complementary RNA nucleotides. Now this antisense strand will complementary base pairing with the other nucleotides. Let's see here. As the RNA polymerase unwinds and opens up and separates these two strands of the DNA, the new nucleotides added up to these complementary base pairing. As you can see here, this is the strand of the messenger RNA 5' to 3'. It means that this strand is the antisense strand, or also known as the template strand. The new nucleotide adds to the 3' end of this strand of the messenger RNA of the RNA. At the end of the RNA, there is the free 3' end. So the free 3' end, so that the other nucleotide can add up to this free 3' end. And the RNA polymerase, as it adds the nucleotide, similarly it moves forward, unwinds these helices, and similarly the new nucleotides keep on binding to this strand, and this is how the elongation of the strand occurs. RNA polymerases join adjacent messenger RNA nucleotides by forming phosphodiester bonds. So these 3' hydroxyl and 5' hydroxyl of the new upcoming nucleotide, it forms the phosphodiester bond. Nucleotides are added to the 3' end of the messenger RNA strand, which grows into 5' to 3' direction. So it will grow in the direction of 5' to 3'. RNA polymerases move along in this direction and continues to unzip the DNA while the messenger RNA is synthesized. As I already explained, that this messenger RNA, this RNA polymerases will move forward. It will unzip this DNA helices, and the new nucleotides will keep on adding on 3' on the 3' of the adjacent nucleotide, and the new nucleotide will be 5'. So 3' and 5', it will form the phosphodiester linkages, but the direction will be 5' to 3'.